Hello, everyone, and welcome to our COVID and the Path Forward webinar. I'm Mark Wisniewski, Senior Director of Biopharmaceuticals at UCLA Technology Development Group. And we're very excited about this panel today, besides our prestigious panelists, but also the salient timing going into the holiday season, and also with, coincidentally, the FDA reviewing the Pfizer COVID vaccine just moments ago. So I'd like to introduce our panelists and turn it over to our moderator. We have Dr. Grace Aldrovandi, Chief Division of Infectious Disease at UCLA Mattel Children's Hospital. Melody Man-Hing Lee, or Dr. Melody Man-Hing Lee, excuse me, Assistant Professor, UCLA Microbiology, Immunology, and Molecular Genetics. Dr. Anne Rimoin, Professor, Department of Epidemiology, UCLA School of Public Health. And now I'd like to turn it over to our moderator, Dr. Paul Krogstad, Professor, Pediatrics and Infectious Disease at UCLA Mattel Children's Hospital.
Good afternoon. I'm very excited to be here today talking to you about recent developments and thinking about the future of COVID-19. As was mentioned, a number of kinds of background and training and expertise, and I hope that you'll find the discussion as illuminating as I felt it was in our brief background, our preparation talk, and realizing that just as progress is being made in the fight against COVID-19 by virtue of having many people coming from different backgrounds, we have a microcosm of that to an extent here at UCLA. A little bit more, we'll give a little bit more of an introduction about ourselves as we go, right at the moment. And so by way of introduction, as was mentioned, I'm a professor in pharmacology and in pediatrics, and I'm a practicing infectious disease specialist for children at the Mattel Children's Hospital here at UCLA. My research career has largely been spent in studying some of the viral diseases that are particularly problematic for children and for society because of their larger scale impact. HIV, enterovirus infections, some research on Zika, and now I'm one of those who have taken advantage of my experience as a virologist to do some work in the laboratory, work in collaboration with others here at UCLA in studying SARS-CoV-2, the virus that's the cause of COVID-19. So I'm going to go around then and just in a similar fashion for people to give a little bit more on where they're coming from, what their expertise is. And I will do so alphabetically, which means Dr. Aldrovandi, I'm coming to you.
Okay, hi, I'm Grace Aldrovandi. As was mentioned, I'm a professor of pediatrics and chief of infectious diseases. My career has predominantly been in HIV, diagnostics, and like everybody else whose world was rocked last spring with SARS-CoV-2, have pivoted to doing studies in this virus.
Very good. So next up is Dr. Lee. Perhaps you could tell us a little bit more about yourself.
Thank you, Paul. Hi, so as mentioned, I'm an assistant professor in the Department of Microbiology, Immunology, and Molecular Genetics here at UCLA. And I'm really fascinated with viruses ever since I was in college, probably because I'm from Hong Kong originally. And Asia always get all the emerging viruses such as avian flu and SARS almost 20 years ago. And my lab mostly studies the host innate immune response, targeting a range of RNA viruses, mostly mosquito-borne RNA viruses such as alphavirus and flavivirus, but recently also SARS-CoV-2 that causes COVID. Thank you.
Very good. And last but not least, by any means, is Professor Anne Rimoin. Please go ahead.
Hi, so I'm Anne Rimoin. I'm a professor of epidemiology in the Fielding School of Public Health and the director of the Center for Global and Immigrant Health. I am a PhD epidemiologist. I've been spending my career working on emerging infectious diseases and immunization programs, disease surveillance globally, the vast majority of my work has been conducted up until COVID-19 in the Democratic Republic of Congo and other places in sub-Saharan Africa, where I've been working on Ebola, monkeypox, understanding hemorrhagic fevers, long-term sequelae to infections such as Ebola, occupational exposures of healthcare workers and other high-risk groups to hemorrhagic fevers and filoviruses. I've also spent a lot of time in my career working with the ministries of health of countries like the Democratic Republic of the Congo, helping them understand, doing serosurveys to understand population immunity to vaccine preventable diseases, risk factors for poor rates of immunization, and understanding in general the epidemiology of vaccine preventable diseases in places like DRC. So since the beginning of this pandemic, I have been working on COVID with Dr. Krogstad and with Dr. Aldrovandi together to look at asymptomatic infection and immunity in healthcare workers and first responders. I've also been running a study looking at occupational exposures to animal workers, animal healthcare, or animal health workers as well. And one of the big things that we've been working on, and certainly the topic of today, is working on vaccine hesitancy in these groups and really understanding what's driving vaccine hesitancy and vaccine acceptance in all of these populations. So that's it.
Thank you, Dr. Moyne. That's you have plenty to keep you busy, obviously. Well, we thought where we would start with the discussion is perhaps where it all begins. You know, what happens at the beginning of infection, and you know, how the body attempts to respond to it, and the problems that it seems like that helps cause. And that is to say, the early immunological response that comes from the so-called innate immune system. And that's where Dr. Lee works. And so Melody, I'd like to ask you to give us your take on what you've learned and where the field is and why this is such an important topic.
Great, thank you, Paul, for that priming. Clearly, that's a very big topic. I'll try my best to sort of cover all the important points. So I guess briefly, as everybody knows, because everyone has been reading so much about it in the news, that SARS-CoV-2 is a positive sense RNA virus. It's part of the coronavirus family. And when it basically infects a cell, what it does is that since it's an RNA genome, it needs to first get in, uncode, and gets translated into viral proteins. And I'm going to come back to it later, but these viral proteins basically are you can imagine all the goodies, right, that the virus brings into a cell so they can copy itself and make more of a cell, because the only goal and the sole purpose of the virus is to make more cells that it can spread throughout the organism, and obviously spread from person to person or organism to organism. And these proteins, turns out that coronavirus also encode a lot of these viral proteins to antagonize the host immune response. So I'll revisit that in just a few minutes. And when the virus has made all these proteins, so express them and so that it can replicate itself, it's going to now get out of the cell and infect new cells. And so that's sort of a very simple view of how the virus infects. So in terms of what we know about sort of the disease and also the host immune response to this virus, so far it's very interesting, I would say, from all the reports looking at the very variable outcomes, right? So I'm sure you also read in the news that it's not a virus like Ebola virus that that I think is fascinating, that stops the world. It's actually a virus that a lot of times that is asymptomatic, right? Like COVID, and causes such a heterogeneous outcome. And so far what we know is that, SARS-CoV-2, just like SARS, obviously attacks the lung and infects the cells in the lung. And so the severe outcome is ARDS, which is acute respiratory distress syndrome, right? That people have to get on ventilators. A very high number, percentage of people go through that, experience ARDS. But at the same time, we've seen reports on cardiac injury, on coagulation, and just very interesting, a wide variety of multi-organ failure, and even reports on neurological symptoms, such as, loss of taste and smell, and more severe forms like Guillain-Barré syndrome, which we've seen actually with the Zika, epidemic. And also, it's really interesting what the long-term consequences of these neurological effects. And again, the question is, does it actually directly affect the central nervous system, which needs to be investigated further. But what is really interesting is this sort of huge variety of disease outcomes that's caused by SARS-CoV-2. I would say the general sort of the current model on the host immune response and how we can really fight it, thinking about modulating the host immune response, is that going back to what I mentioned earlier, the virus, this virus is amazing. First of all, it's huge for an RNA virus. And so it can encode lots of goodies like the viral proteins I mentioned earlier. And a lot of them can antagonize the host innate immune response, mostly the interferon response, which is the first line of defense against this virus. And so as a result of it, you can imagine that people that are infected initially during the acute phase, they're not able to mount a very effective antiviral response. And so the virus basically has some advantage, right? Replicating more of itself. But at the same time, we're seeing a huge inflammatory sort of outcome, a lot of times due to recruitment of basically inflammatory macrophages to the site of infection. And so I think the current view on sort of thinking about all this is that if we can try to enhance the host immune response early on so that we can we would prevent the virus from or make the virus less effective at antagonizing the host, but then later on time it and suppress the inflammatory response. That's probably going to give us a better outcome, although you can imagine how complicated the timing of things are. And I'm sure, the MDs on this panel, Paul and Grace, can tell us more about all those therapeutic options. Yeah, I think that's pretty much sort of a summary of all this.
Not to get too granular, but I thought of one follow-up question while you were speaking, and that has to do with what the medical community and scientific community have heard, as well as what the public has heard a great deal about. The fact that the question of how long protection might exist, if at all, after an initial infection. Can you comment on how, what you study, innate immune responses and the virus's antiviral mechanisms might, the mechanisms that have been proposed for how the virus sort of interferes with having permanent immunity the way we generally do after measles, for example?
Yeah, Paul, that's a great question. And obviously, for measles vaccines, even though getting the natural infection will give you lifelong protection, you don't want to get naturally infected by measles virus, because now we know there's long-term consequences of our immunological memory being erased right after a natural measles infection. And I think this is sort of coming back to thinking about how coronaviruses, these are master manipulators of the host immune response, specifically the interferon response, because studies have been done on SARS and MERS, right? The previous pandemic or epidemic coronaviruses, that each of these viral proteins can specifically target distinct steps in the interferon pathway, so that either the virus cannot be sensed or the signaling components are not being activated. And so you can imagine that probably the consequences of that is, you might also have a weakened adaptive immune response, because clearly the interferon response or the host innate immunity is tightly correlated with activation of the adaptive immunity. And so I think what we're seeing with the studies, for example, and I'm not expert on T cell and B cell biology, but for example, there are reports on T cells being sort of activated in persistent COVID disease that leads to T cells exhaustion. So I can imagine how that could be an issue, whether you're symptomatic perhaps or not, that it could affect your outcome to other viral infections. So clearly, again, vaccine is the way to go because you can design the vaccine in a way that you can trigger these appropriate immune responses that perhaps the virus is so, is so ingeniously designed to antagonize them that you wouldn't be able to get the full benefits of protection from a viral infection versus a vaccination.
Yes. Well, in fact, so with that, I think we'll try and shift our discussion a little bit towards vaccines, and I was going to bring out a couple of things with Dr. Aldrovandi. Melody raised the point that vaccines do much more than you actually think. The measles vaccine doesn't just protect us against it doesn't just do what it's designed to do, which is to prevent measles disease, but it also keeps us from losing immunity during childhood to other viral infections, or even keeps us from losing the effects of our prior exposures to other pathogens and prior exposures to other vaccines. So that's a big ask, and yet that vaccine does it. We're still at the point of focusing on just trying to stop, to slow the spread of this condition and by giving people protection. You more than most people I know, read the New York Times faithfully, and I'll bet a good number of the 144 current participants know a lot about the vaccines that are out there being studied because of reading, these newspapers that really drill deep and give you a lot of information. So, people will have heard of the two that are now approaching approval in the U.S. and others. Perhaps you could can you do a little bit of the overview, Grace, and talk about the categories of vaccines and how they're coming towards us, how they're about to be something that can be used as a tool in this pandemic.
That's very happy to, Paul. And I would just like to expand on what you and Melody were talking about. Everybody thinks, oh, well, natural infection is better than a vaccine. And you've mentioned measles. But I'd also point out that, we all get chicken pox. And then when you're older, you get zoster. And the natural infection doesn't keep you from getting zoster. So for older folks, you'll know you get Shingrix. That's an example where the vaccine is better than natural infection. There's also the human papilloma virus that prevents cancer. That vaccine is better than natural infection. In young kids, Haemophilus influenza type B, the vaccine is better than natural infection. Tetanus, you can end up intubated in an ICU for months. And after you recover, because we support you, you won't have enough antibody to prevent you from getting tetanus. So point number one, natural infection isn't always better. I would like to start off by saying, can we just take a second to thank science? I mean, this is extraordinary, all right? We didn't realize this pathogen was in the human population till December a year ago. We didn't have the sequence until January 10th. By January 15th, the VRC, the Vaccine Research Center at NIH, had already designed an mRNA construct for Moderna to take forward. And this wasn't, people think, oh, they've just pulled it all out. All the science that we have is really based in a tremendous infrastructure that we have built primarily through the NIH, but through others in this country. So they learned from RSV, a respiratory syncytial virus, how to optimally design this. And now we have two vaccines that look like they're 95% effective. Okay, that is like a home run. you're winning the ball game. Nobody could have anticipated this. And so we did it because science is awesome. Scientists are great and have worked 7 days a week, 24 hours a day for the past year to get this out. And it's not only scientists, it's regulators, okay? FDA is turning around things very, very fast. People have participated. The Pfizer trial enrolled 40,000 people in 6 weeks, okay? That is huge. So we have really all gotten together and we have shared. Never in my scientific career have I seen such transparency and sharing of reagents. So I think, you know, there's a lot of bad news, but I think there's a tremendous amount to celebrate. So to answer your question, sorry for that preamble, but it is really, really exciting times. And it's so hopeful, even though we're going to be told, you know, over 3,000 people are dying, and it's only going to get worse over the next few months. We're being asked to stay away from our loved ones during Christmas, but the cavalry is coming, and the cavalry is a scientific community getting together with folks and coming up with this awesome, awesome vaccines. So you asked about the vaccines. Well, in this country, though there's across the planet, there's about 40 different vaccines. In the US, we have 6 vaccines that are in trials. And as you mentioned, and I'm sure everybody's heard, the first vaccines that are going to be up to the FDA for EUA are what are called mRNA vaccines. This is a new way of doing vaccination, and I'll talk a bit about that more. Traditionally for vaccines, we've used live attenuated, so weakened forms of the pathogen, like with to induce immunity, or we've given you the protein. These mRNA vaccines are relatively new. There's been some studies in cancer and other things, but the big advantage is that they're really fast. So what is mRNA? mRNA is basically the blueprint for your body, for your cells to make a protein. And what scientists have figured out to do is if you put this mRNA, which is incredibly labile, if you work in the lab, you have to wear gloves, it just dies, in a lipid particle, and they inject it into you, and it's taken up by the cells. And then it uses, and then once it's inside the cytoplasm of the cell, your machinery there says, oh, this is a blueprint, I'm going to make it. So your own cell is making the protein. It's not a protein that we have made outside and are injecting into you. I would emphasize that it stays in the it stays in the cytoplasm. It doesn't go into the nucleus, because some people feel that, ooh, it's a gene, they're going to alter my genetic composition.
Right. It's not a gene transfer.
It's not a gene transfer. It's really hard to get things into the to the nucleus, and it's worked like gangbusters. So you know, but there are issues, and people are a bit concerned, understandably, that, you know, this is too new. All right. But again, I would say that the mRNA only lasts 36 hours. You know, obviously we don't have a lot of data, but we have some data. There's no evidence that it gets into the circulation. The other thing people worry about, well, this is just too fast. You know, I know that it takes 10 years to develop a vaccine, and people have moved really fast. They've moved through the preclinical and the animals, and the FDA has been inspecting the factories that make these vaccines before the companies have gone to them. The governments have bought the vaccines at risk before we knew that they worked. They bought them so there could be production for them. And, you know, so that part's gone fast, but the proof of a vaccine pudding, you know, the one thing that we use to assess safety and efficacy, that has not been shortcutted. And that is your phase 3 clinical trial, where half the people get a placebo. In the case of the Pfizer mRNA vaccine, they got saline, they got salt water injected into them, and the other half got the vaccine. And the data are spectacular. It's 95% effective. After the second dose, there were 162 people in the placebo that got COVID, and only 8 in the vaccine arm.
And which study are you talking about there?
This is Pfizer's.
This is Pfizer's.
Pfizer's mRNA. Moderna, they've only had, it's science by press release, but, Pfizer came out today in the New England Journal of Medicine. They include the protocol. They include all the appendices. I suggest that everybody look at it. In addition, 2 days ago, the FDA on their website, they have a 58-page document summarizing all the data that they got from Pfizer. But this is just tremendous. And just before this call, the Vaccine and Related Biologic Products Advisory Committee voted to approve the Pfizer vaccine. So it's
Bear in mind,
tremendous.
and it hasn't been approved yet, right?
No, it hasn't been approved. And then tomorrow the ACIP will look at it, but I would expect that it will be approved shortly.
Right.
So,
I'm glad that you brought this up, that what has not been shortcut at all is the need to observe for fairly long periods of time for adverse effects. And that monitoring is going to continue for quite some time. But in the short term, the months that people have been followed after these volunteers were immunized, no safety signature emerged that was in any way worrisome. We've now heard, of course, about maybe having some rare hypersensitivity events with one of the RNA vaccines.
Right. So the Pfizer vaccine was released in the UK, and on December 8th, the first day, there was a case of severe allergic reaction. I would point out that if you look in the exclusion criteria of the Pfizer study, they excluded people who had severe anaphylactic reactions to vaccine components. So we'll have to see. There's two people, and the UK has issued an advisory that if you have severe allergic reactions, that you shouldn't get the vaccine. And that's something that's going to be investigated, because we're going to have to try to understand, who is more at risk at getting this serious adverse event. But I would say that, compared to the flu vaccine that people get, that this has more reactogenicity. All right. So, almost everybody, over 80% of people are going to have a sore arm for a day or two. Over half the people who get the vaccine are going to have headache and be fatigued. About a third of people are going to have chills, and 15% will have fever. Now, this is not a bad thing. Think about it. This is your body's immune system seeing this protein and marshaling all the parts of the immune system that Melody was talking about to generate the cellular and humoral and innate responses to fight the bad guy. So if you have a trainer, they say, no pain, no gain. Think about these reactogenicities, these side effects, as, your body seeing the bad guy and marshaling all its resources. But it's not going like, your usual flu vaccine, but it's much more effective. And I would point out, like people say, well, why are they getting an EUA? We know from all the vaccines that we've studied that most of the uncommon clinically significant side effects are seen within the first 6 weeks. And so that's why the FDA has mandated that you can't go to them for an EUA before you have 2 months of follow-up. But so it's not the numbers or the rigor of the science that's led to an EUA, an emergency use authorization, as opposed to the BLA, which is like the full authorization, full approval. And that's just because we only have 2 months. But today on the committee call, Pfizer announced that it will be applying for a BLA in April. So as things evolve and we get more data, we will have full approval.
Okay. Just one other, we'll come back to some discussions about the vaccines themselves in a few minutes. There are of course, other variations on a theme, other ways in which people are trying to make the viral spike protein be presented to the immune system so that you can get an immune response. It seems that that's the dominant strategy. The other approach is to use a vaccine, use a virus that has had a gene removed and then has had the gene for the spike protein inserted so that it can be turned on briefly. Any initial thoughts about the impact? The initial immunogenicity data has looked good with several of these, and China has even approved its own. Russia has approved its own that follow this strategy. Any thoughts about the relative comparisons here? With the versions that are being of this strategy are being tested in the United States.
No, so as you mentioned, there are these the adenoviral vector AstraZeneca, Oxford AstraZeneca's vaccine will is close to applying for an EUA. As you've mentioned, China, Russia, and United Arab Emirates yesterday approved
Yes.
the Chinese vaccines. They all look remarkably good in terms of getting you antibody levels, but they do have advantages in that they're going to be much cheaper than these mRNA vaccines. And you've probably heard that these mRNA vaccines, because they're so labile, and in particular the Pfizer vaccine, has to be in a very deep freezer, minus 80 Celsius. And so logistically, it's very difficult to give out. So I think, we've got to immunize the whole world. And I think that having multiple vaccine candidates will be helpful, not only to reach everybody on the planet, but also it may be that, certain vaccines work better in certain populations. And we will, I'm sure there'll be comparisons and we'll learn that relatively fast.
I want to make one more comment and then ask you a question about the vote, because I know you were watching that today. The comment I was going to just make is that the RNA vaccine strategies are appealing in one way because, the purity of the system that you're looking at, that it's RNA packaged with, proprietary mechanism, there's a proprietary approach to packaging an mRNA that's designed to express this one and only one viral protein, whereas with an adenovirus vaccine, you're actually using a virus to begin to generate an immune response. So it's difficult to, one is sort of simpler, one is more difficult, that is to say the viral vector approach. But I can't help but think that if we continue to see, if the RNA vaccines work as well as those initial immunogenicity studies suggest, and the initial clinical data that trumps, that really makes it possible to think about these being approved. If those work, then that's going to be a platform that can be retooled. This is our third pandemic with a coronavirus in the 21st century already. And we would be foolish to think there aren't other potential pandemic viral infections coming. Having something we could retool fairly quickly to make a functional vaccine, I think that's an advantage to it. And I know we're getting some questions already, and I'll just toss one to you before we move on to another topic in our discussion this afternoon. One of the listeners asked, were there any issues that led to the no votes besides the 16 to 17-year-old inclusions? And I haven't read through all the safety reports, but this person also says, what of the Bell's palsy report? Did you look through that lengthy document and see the comment about Bell's palsy? Is it a single case or?
Yeah, no, the Bell's palsies, there were 4 cases of Bell's palsy in the vaccine group and 0 in the placebo. That doesn't, the FDA said that isn't really above background, but obviously it's something that they're going to continue to follow. And, we were going to be immunizing so many people all over the world. I think we will get more data. There was discussion And the issue of whether there were enough 16 and 17-year-olds to have the indication go to that age. Just based on the discussions, it was clear that a couple of the people, of the 4 people, so the vote was 17 for approval, and the resolution was based on the totality of data, the safety and efficacy profile warrant going ahead from 16 and up. I'm paraphrasing, I don't have the exact language. There were a number of speakers who didn't feel there were enough 16 and 17-year-olds in the trial, and the risk of severe COVID disease in that age group wasn't enough to warrant it. So, but I don't know how many of the 4 that voted against, that was their reason. You know, there was a lot of discussion. That was a major thing. You know, there's still a lot that we need to know about this, because if you look at the Pfizer study, there were about 10% African Americans. There were 20% over 65. And, you know, there were only 153 16 to 18-year-olds. There were very few Native indigenous people in the study. You know, not a lot of people over 85. So, you know, we have a lot more to learn about it. And as I said earlier, it may be that certain vaccines are better in certain populations, but given the high death rates in older people, also Pfizer didn't include nursing home folks who are prioritized for it.
That might have other comorbidities, other illnesses.
Yeah, but 45% of the people in the trial had other comorbidities, and 35% were obese, a BMI of over 30. So I think for the common comorbidities, there are a fair number of people. You know, the big gap is pregnant and lactating women and children.
Yes.
And immunocompromised people. Now, in the second phase, Pfizer allowed people who were HIV infected, hepatitis B infected, and hepatitis C infected to enter the trial. And I don't know offhand the numbers of those, but it all looked very good in those. So there's still a lot to learn. I don't think we know everything, but it really does look good at this point.
And I have to agree with you. And, you know, I'll try to, we're going to slightly change direction for a few minutes. I want to go all the way back to where you started with this. You've just finished commenting on things that we need to know, and bearing in mind that it's years of experience. It's much larger groups than the 21,000, for example, that were in the vaccine group in the Pfizer study. It's much larger numbers that show you the totality of the strengths and weaknesses of any pharmaceutical agent, including vaccines. But you know, this is where we've managed to get in an incredibly short period of time. Part of what's probably accelerating this compared to, let's say, the Salk polio vaccine was that we have the data transfer abilities that we didn't have with any vaccine before. Nothing has gone as quickly for a large scale vaccine study as this has. And I'm glad you gave credit to people like the FDA. Our regulatory boards here at UCLA, for example, have also just have really put out extreme effort so that things could move absolutely as fast as possible, not waiting for the next scheduled meeting of 2 weeks down the road when work needed to be done. It has been a high priority. And you know, I want to switch briefly, you know, to talk just a little bit about what else we've learned that is making a huge difference. When this started, there weren't, when the pandemic started, we heard about how they were managing patients in China and throughout the world. Essentially, some experimentation was done to try and figure out how this illness is different than others. There was a lot of hypothesis generation just based on how some patients did and with one treatment and some another. And there's just any number of directions you could talk about the innovation and the speed at which other medical science has come along. Finding the right way to use mechanical ventilation to help people who've gone into respiratory failure. When is it time to move beyond the high flow oxygen support that is possible, and having to move forward to some sort of advanced support, mechanical intubation, being connected to a ventilator, taking the most extreme step of essentially connecting a person to a heart-lung machine, taking the blood out of the body, connecting it to the same machine that is used to keep people alive during open heart surgery, putting oxygen into that blood and returning it to the body through all this tubing, through all these machines without causing a blood clot. You know, that's technology, that's among the best examples of 20th century medical technology, but now being applied to an explosive pandemic. And, you know, with people surviving with these modalities, we didn't know what would work. People didn't know what would work. And sometimes there have been things as simple as realizing that a measure that's been used sometimes for other disorders, putting a person in prone position, face down position, that or on their side, you know, the positioning of an individual based on the may help improve the actual transfer of oxygen into blood. Those very basic clinical points, let alone learning the importance of anticoagulation, coming to find out very quickly how it's not just big blood clots, like big blood clots to the lungs that can block a large vessel. It's having small vessel disease that can damage your kidneys or your brain, and as part of the lung disease. Just so much has been known. You know, you and I, Grace, we haven't been flooded with pediatric patients with COVID-19. We have had some here at UCLA, but nationally, the number, of course, is relatively small relative to all of those of even young adults. But still, there's had to be all this progress in understanding not only how to manage what we were immediately seeing, but then catching up with new this one known as multisystem inflammatory syndrome in children, MIS-C, and learning how to adapt, learning how dangerous it is and isn't, finding out what you need to do and what you don't need to do. Truly amazing. I guess.
Yeah, I really would like to give a shout out to the first line healthcare workers who have literally put their lives on the line, and even, you know, in a sense, the lives of their families to take care of really, really sick patients, you know, working incredible hours and really, you know, building this spaceship as you're trying to fly it. You know, people say it's a plane, but when you look at how, you know, New York and other places have just been overwhelmed with the number of patients. And I don't think that, you know, we can express our gratitude to really people who are heroes. And it's the nurses, the doctors, the respiratory therapists, the lift teams that are moving these patients. And it's been really hard. And I think, again, when we turn to, you know, the bedrock of science, which is randomized clinical controlled trials, which people are doing under extraordinary conditions, that we learn what works and what doesn't work. Right. And, you know, so we've learned hydroxychloroquine is actually bad. All right. Yeah. But, you know, steroids are good. And we know we've adapted our clinical trial design, and also right. People have been sharing at an unprecedented rate. I mean, 7 days a week, there are major journals are making their COVID publications available online without a paywall. That's huge, all right? And so that has really enabled us to make the progress that we have.
I wanted to get Anne involved here so we can talk about the bigger scope of this. But I want to close just with commenting how impressed, as you have been, the frontline practitioners, the emergency room physicians, the nurses who go, you and I, when we see patients, go in once for a period of time, then we leave and we might come back later the same day. A nurse who's going in the room every 5 minutes sometimes just to to keep a person stable in the ICU or maybe parked right there for hours. They take the brunt of the risk. I've been so impressed, even at the care they provided for me, the times that in my involvement in helping enroll people for clinical trials, I've been in ICUs, that they're looking out even for the people around them, the respiratory therapists, and so on. This has just been an impressive mammoth effort. Now, but I do want to ask a little bit of Anne about how she, Professor Anne Rimoin, about, are there things we're not yet thinking of that we should be wary of? And this brings up the fact that, that your work in Africa, especially in following and seeing the impact of Ebola immediately and then coming in later, how that makes you think about what we're currently facing. We're very much in the moment just trying to get through, but are things that we should be vigilant for coming up based on your experience and seeing how the long-term effects, for example, of Ebola infection?
Well, thanks. I'm really glad that we have Grace here today because this is so of the moment. So I'm going to make my answers as short as possible because I really do think for everybody, we're getting literally breaking news here. And this is really exciting and a perfect example of these webinars or these kinds of things. They hit at the right moment. We have all of you who really have this expertise, and the vaccine is the of the moment question. So I'm going to just say a few very quick things, and then I think we should get back to asking questions about this vaccine and the approvals. So I would say, from my experience working in pandemics, there are a number of things that I was, this is not the first pandemic. It's not the last. We're going to be seeing more of this. We have to get better at doing this kind of work. And we need to be able to move quickly with research. And I think, that was one of the things when actually the three of us were at the very beginning of this talking, I was used to working in a place like DRC where you have to pivot quickly and you're ready for, I work in epidemics and I work in outbreaks and I'm ready to just throw things at the wall and get started as quickly as humanly possible. The things that we've been missing in the past are the kind of research infrastructure and the funding to be able to quickly jump on a question and answer it when you need to jump on it, which is immediately, not waiting months and months and going through NIH going through, study sections and waiting for reviews. And that was one of the things that we as a team did just, I think, so uniquely is that we jumped on this. We raised money to be able to start answering questions and we got right in there. And I think that is so critical. And going forward, I really hope that there's going to be funding available for these kind of rapid research and responses, because that is the only way to be able to do it. And you need to plan for these kinds of things before it happens, not once it's going. So I think that that's something that's super important. We did the same thing with Ebola. I'd been working with Ebola survivors for many years in DRC. I'd been doing studies on immunization, had the ability to do serosurveys and collect PBMCs, collect samples from people. And so the minute that there was an Ebola outbreak, what we were able to do was use funding, because the Gates Foundation was very willing to let us pivot and move directly into collecting samples and looking at immunogenicity of the Ebola vaccine in DRC, understanding occupational exposures and those kinds of things. One of the things we learned from the Ebola outbreak is that there are long-term consequences of having these viruses. We know people in DRC and in other places, that there are longer term effects like renal failure, mental, these neurological sequelae, lots of things that can happen to you. And we learned that. We started looking at it in other viruses as well. And that's why I think we're learning now what these consequences are. It would not surprise me that going forward, we're going to find that there are consequences to any infection that you have. And just much like we look at epigenetics now, that things that happen to you can in fact be integrated into your genetic code going forward, it makes sense that things that you are exposed to can shape you in ways that we hadn't really anticipated. So because I'm not a clinician, I'm not going to get into the details of it, but I would say that we look at things, and in terms of long-term sequelae, it's, I quote Yogi Berra whenever possible, but it ain't over till it's over. And that we really understand that this, you're going to carry forward exposures and experiences experiences that you've had, and that it will probably have longer impacts on your health. I would say that, you know, so I would say being very, very quick in terms of being able to respond, looking at short-term and long-term effects, really understanding what's happening in real time, planning for the future. You know, my hope is that there will be people studying this for a long time to come and seeing what's going to happen in the future. And, you know, I think that, you know, in those, what will we see going forward? We will see more viruses spreading into human populations. There's no reason that we will not see that. And we just need to be, you know, I think we understand now how broken our system is and how we do need to do better. And we need to really shore up all of the foundation of our house so that we can have a solid foundation from which we can move going forward.
Yes.
I actually have a question. I wanted to just get back to Grace for a second. Sorry, Grace, I'm going to ask you the question just because this has been, I think that this is really important when it comes to the vaccine. We've talked about all of the reasons why people did not, you know, why there was some question about the approvals. How would you go about getting the, how would you go up shoring up these gaps in the data and understanding maybe testing in children, testing in pregnant women, testing the vaccine in older populations? How does that happen and who does that?
Well, that's already been planned by, you know, Pfizer, Moderna, all the vaccine companies have plans to go into children and pregnant and lactating women. Just yesterday, Moderna announced it's going to have a 3,000 child study. And in fact, in the United States, you have to do studies in children. So it's going to be coming to the newspaper and an FDA commission near you. And I think the issue of pregnancy is one that's particularly important. 70% of healthcare workers are women. And actually in the Pfizer trial, there were 73, even though they tested you to see if you were pregnant, 23 women in the study became pregnant, 12 in the vaccine, 11 in the placebo, if I remember correctly. 2 women in the placebo group, one had a miscarriage, the other another adverse effect. We don't know in the vaccine. So obviously this is something we have to take very seriously and study really well. But I will say the CDC, the FDA, the World Health Organization, all the countries that are approving these vaccines are committed to sharing all the data in real time. There are multiple platforms for reporting adverse effects to vaccinations in this country and across the world. And in fact, they've even, you know, I always ask, is there an app for that? Well, there's an app for reporting side effects when you get it. So I think that we're going to continue to see a lot of data, and we're going to be sharing with it, you know, not only with regulators, but with the public, which I think is really important. Vaccines don't save lives, vaccinations save lives. So we have to get people to take the vaccine.
And yet
Well, I can actually tell you right now, I'm getting texts right now from our team who's analyzing our study on vaccine hesitancy. And we're finding that between 2 months ago, or from the end of October to now, we've gone from having 66% of our healthcare workers saying that they were not willing to get the vaccine as soon as possible to only 32% saying that they wouldn't be willing to get the vaccine. So, I mean, I think that this process has made a very big difference.
Oh, of course, we have to put in the usual proviso, or Tony Fauci, the ghost of Tony Fauci, will come back to get us at some point. It isn't just about vaccines that washing our hands, keeping distance, and wearing masks are all measures we need to continue doing. A vaccine won't take away their importance, and you know, even after a person's had disease. So far, since there have already been examples of second cases occurring, it means that, you know, somebody who has already had a case also needs to be wearing a vaccine, wearing a mask, because the disease itself doesn't provide permanent protection.
Yeah, I think a mask is not a vaccine is not a passport not to wear a mask.
Yeah.
And the one thing we don't know about these mRNA vaccines, the endpoint was symptomatic disease. We don't know if vaccine recipients had virus in their nose and they could have spread it to somebody. So just because you're vaccinated doesn't mean you should go hug grandma.
Right, which is, resuming some of our more normal social interactions, including close physical contact when you meet people you haven't seen for a while. We're going to have more information to be able to answer the question to what extent is a vaccine protective even against transmission.
And we're going to have 75, 80% of the population infected. And as an Italian, not being able to hug and see Annie and you really kills me. But I will continue once I'm vaccinated.
Melody, there was a question that you wanted to respond to as well, and that was, I guess about the persistence of this virus or along those lines. Do you want to address that? We saw that come up.
Yeah, thank you, Paul. No, I think that's an excellent question. And I wanted to, answer live to that attendee. And I guess this is also tying to your earlier point, Paul, about, the fact that now we have 3 coronaviruses that cause pandemics. How can we better predict, right? That's really the billion-dollar question for the next pandemic. What will be the best strategy? And it's sort of a provocative idea I want to throw out there to the audience, is that the 2 reasons why Asia has been one of the places with all these sort of emerging viruses is one is reservoirs, viral reservoirs, meaning that there is animal hosts that harbors a tremendous diversity of these viruses. And they're persistently infected, but they don't get sick, or not, they don't have severe disease. And so they can carry this virus for a long period of time, and they can transmit the virus to intermediate host. I'm sure you guys have all heard about, the host for this SARS-CoV-2 is the very cute animal. Now the pangolin, I forgot the name, but it's the cutest animal. And we know that for SARS, it was thought to be sort of the raccoon type animals, the civet cats, right? The palm civets. And so one reason why Asia is so prone to it is because there are reservoir hosts. In the case for coronaviruses are bats that carry just all this diversity, which can lead to cross-species transmissions, which makes it so hard also to predict the next pandemic because there's so many possibilities. The other reason is because in Asia, a lot of people, on the farm, really, we live very closely with our animals. And so, for example, for flu's reservoir is actually in aquatic birds. So those birds don't get sick. And actually the virus replicates in the gut in those aquatic birds. However, the virus can jump from these aquatic birds to livestock, right? So basically in the chickens or the pigs, and that's where we get the next huge pandemic, because all these viral genetic material can reassort, meaning that mix and match to to give rise to the next scary new virus. So I think there's a lot of interesting thing about just the ecology and the biology of these viruses that make these viruses so hard to predict, right? What is going to be the next one? So I guess the provocative thought I want to throw out there is, in addition to what Paul was saying with the mRNA vaccine being easy to manipulate, right, when the next pandemic comes. The other thing is to think about how can we better design these vaccines? What do you put on these vaccines? And one idea I thought, and obviously it's very hard to carry out because there's so many possibilities, is that can we actually generate vaccine candidates already that are more the viral strains that are carried by these reservoirs? Because eventually these reservoir strains are going to be the ones that make the jump and cause the next pandemic. But the logistics of it all, clearly that's a very tough question. But
yeah, I think there is one thing, in fact, that we have done wrong, over, let's say, the last 20 years, in terms of the results, I think, from the dearth of funding, that people have, and that is that we didn't learn from the previous experiences. Ebola outbreaks go back go into the last quarter of recognized Ebola outbreaks, going to the last quarter of the 20th century. Efforts were made to develop these vaccines, and after a point in time, things seem to sell out. And I'm sure that it has to do with the fact that there simply wasn't.
Sorry, Paul. I just wanted to thank Dr. Ramon for her participation, because I know she's saying she has to drop off right now.
Yeah.
But if the other panelists are available, we can continue the discussion for another 10 minutes.
Okay.
I wish I could stay. Thank you so much for including me. I think that this is such an important discussion, and we really need hours and hours to continue it. I look forward to more opportunities. Bye, everyone.
Thank you. Thank you again for your participation, Dr. Ramon. Sorry, Paul.
Oh, that's all right. Long story short is that there were candidate Ebola vaccines, and progress was slowed for a number of years, simply because I think there wasn't enough eye on the ball, keeping funding going to try and bring that to fruition, to bring a functional vaccine. Now we have an FDA-approved vaccine after the 2014 Western Africa multi-country Ebola outbreak, and it's a very good vaccine. But the groundwork for it had been laid. In a similar fashion, I hope that we don't take our eye off of future pandemics that may come our way from coronaviruses, which, as Dr. Lee has said, these are big viruses. They have the ability, they recombine, mix and match pieces of their genomes more than any other virus with a similar RNA genome, using RNA instead of DNA to hold your genetic information.
Yeah.
We have to pay attention.
Well, I think we have to continue funding. And I know of preeminent coronavirus experts 5-7 years ago were really concerned that they were going to lose all their funding because,
Yeah.
at first, before SARS-1, coronaviruses just give you colds. Why do you study them? And then there was MERS, and we had 10 years pass, and then MERS, but that's only in old men with camels, right? And so there, study sections, and I think we really, really have to give a shout out to Tony Fauci's leadership and his commitment to continue funding. And we're talking about coronaviruses now. We would have guessed that it was flu for a variety of reasons, but there's Nipah viruses. So the reservoirs that Melody is talking about, are real. Bats are just amazing. They harbor tons of different viruses that once they get to us or through an intermediate host can cause a lot of badness. And I think shutting down the world economy, killing, so many people, infecting billions, that I think we should all go away and realize, and this isn't just a conflict of interest, research saves lives. And we have to continue to funding research at all levels, not just clinical research, but basic research. We wouldn't be able to make these mRNAs unless we had really good people understanding lipids and manipulation of genetic material.
And let's see, if we're finishing 5 minutes, here's a question I think that is particularly bite-sized. That is to say, here it is. And that's that if a vaccinated person can still transmit, how does that affect the timing or ability to achieve herd immunity? At least one of my responses is there's shedding and then there's shedding. That is to say, some people shed large amounts for very long periods of time. We see that in some of the common congenital infections in children, cytomegalovirus shedding and mumps being shed in the rubella being shed in the urine for a couple of years. Then, in contrast, you have the sort of shedding that was seen with polio and oral polio vaccine, where now you're back to months, and immune responses do hurt of viruses. And so it would be expected that they would decrease if protective antibody makes its way in the nasal passages. You would predict that the amount of virus that's there would decrease, and that is something we've learned from influenza as a factor in the ability to transmit.
But
So I think it
It's
can
important-
matter. What do you think, Grace?
Yeah, I think it's important to point out that we will get those data. If I understand Moderna's studies, they are actually doing nasal swabs weekly. Pfizer's going back to analyze, and the CDC already has plans in place to monitor folks to see if they're shedding. There are animal data to suggest that, yes, it will decrease, and we may even have sterilizing immunity, meaning that you won't be able to get infected. 3 months from now, I'm pretty sure we will have an answer for that. But you're right, we don't need to have a complete breakdown, but we do need to have people take up vaccines to stop this.
Right, especially because we, with all of our failings, have had such a, with all of our limitations as people, have not been able to even consistently apply the public health measures that would have made a much greater difference. Well, we've reached our time, the time that we talked about, and so unless there are any final points that you would like to make, we'll go back to our hosts and thank them for the opportunity to be here today. So personally, I want to thank you for the chance to talk about this, this period of incredible difficulty and strain, but something that certainly I never thought would really happen in my lifetime. I never really envisioned running into something quite like this. And I'm happy to have a chance to talk about, my concerns, my considerations, and my thanks for the people that have done so much locally, nationally, internationally to try and help address this pandemic.
Yeah, and I'd like to truly thank, everyone on the panel for, the timing, the insight, the engaging discussion, the different perspectives. And going into the holiday season, it's real hope, and, to quote, the risk of the nightmare before Christmas, balance of, and also the opportunity potentially to bring you back in 3 months to say, well, what does success look like now that there's a rollout? What are the challenges? Things of that nature. But again, thank you. This has been very, very enjoyable, valuable. And again, we appreciate your time.
Well,
Thank you so much, Mark, for the invite.
Thank you.
And thank you, Paul and Grace. I learned a lot.
Thank you, everyone. It was brilliant.
Stay home.
Yes, next time we'll plan to go all night. Great.
all right.
Thanks, everyone.
Thank you. Bye-bye.